Hello, and thank you for joining us for this Capsugel Dosage Form Solutions webinar entitled Biologic Comparison of a Novel Inhaled Insulin DPI Formulation to Exubera. We're going to discuss capsule and formulation innovation for inhaled dry powders, and in addition highlight a partnership between Capsugel Bend and Loveless Respiratory Research Institute. My name is David Bodak, and I'm a Senior Director of Pharmaceutical R&D at Capsugel. I'll be moderating and presenting part of the webinar today, and my colleague, Dr. Philip Kuhl, is a research scientist at Loveless Respiratory Research Institute, will also be presenting and highlighting some of the work that was done at Loveless. Over the next 45 minutes, we'll discuss important aspects of formulation process development for an inhaled biotherapeutic, highlighting the synergy of our capabilities, and provide an overview of a recent case study comparing the inhalation performance of a novel spray-dried powder of insulin to Exubera. We'll then have about 10 minutes or so to answer any questions from the audience. When you have a question, please submit them at any time using the Ask a Question tab on the left of the screen. Feel free to use the tab at any time. If time allows, we'll answer your questions at the end. If not, we will follow up later offline so that we're responsive to any and all questions that we get. Finally, at the end of the webinar, we'll have a brief survey. We appreciate your participation in that, as it helps guide and make these webinars that we have more useful to what people are interested in seeing. So we look forward to your questions, comments, and suggestions. With that, let's get started. So again, we're here to highlight a partnership between Capsugel Bend and Loveless, and we're using a case study that was published in AAPS PharmSci Tech in 2014, where we took as a model insulin and developed a dry powder, and then compared that using some pretty sophisticated, interesting animal models that were developed at Loveless, comparing what we developed with what was a commercial product at the time, just to kind of show similarities. Initially, Phil and I will give a brief update on both of Bend Capsugel and Loveless to talk about our capabilities and anything new out there, and kind of use that to show the real synergy of how the two companies are working together to streamline inhaled drug delivery, inhaled drug development. Once we get that out of the way, then we'll dive into the details of the study. And so to get started, we'll just, as the brief update and discussion of the capabilities that Capsugel Bend, start with, Capsugel Dosage Form Solutions offers a wide range of formulation design, development, and manufacturing options. Today we're going to specifically be talking about the capability and expertise in developing dry powders for inhalation, and somewhat around the types of formulations that are amenable to stabilizing biotherapeutics. But just so everyone's aware, we also do quite a bit of work in the areas of bioavailability enhancement, looking at spray-dried dispersions, hot melt extrusions, liquid-based formulations, and some nanocrystal technologies. And then using those and other platforms around multi-particulates for modified and targeted release, which also includes things like taste masking for pediatrics. And of course, taking into account how all of those formulations interact with the dosage form, and specifically some of the capsule-based options. So again, today we're going to be focusing mostly on the dry powders for inhalations area. And when we talk about Capsugel Dosage Form Solutions, it's useful to present a little bit around the business model of where we think we are have the most expertise in developing drugs with partners. And the business model really is to be in the specific areas of technology that we have, to be encompass the entire train of interacting with the client and their target product profile to design, develop, and manufacture into a finished product. And that integrated approach with the feedback from the client and the collaboration is really important to us, and how that overlays across the different technology platforms. And I think the relationship that we have with Loveless highlights how important it is to have the right collaborations in order to enable this. So the animal models and the expertise that Loveless brings to the table really has good synergy with being able to, on the front end, understand what's important about the formulation that you're trying to develop. And Phil will take us through that in more detail later. Briefly, a slide to just highlight Capsugel's global footprint and where Dosage Form Solutions resides within that global footprint. And I'm speaking to you here from our site in Bend, Oregon, where we do R&D as well as manufacturing and spray drying. There are sites across the world, including other sites in Europe and manufacturing sites elsewhere. And now just a little bit specifically about the facilities that we have here at the Bend site. And along the lines of the design, develop, manufacture mindset, we're vertically integrated to handle exactly that at each site where we perform these functions. And here in Bend, we have R&D labs where we have expertise in the physical science, analytical chemistry, essentially how do we develop these formulations and we test them and we ensure that they're functioning the way they were intended. We have an engineering R&D plant where we reduce things to practice, so new technologies or formulation concepts get reduced to practice and scaled up. And then we have the infrastructure on site, on campus, where we can do the cGMP manufacturing of those formulations up through phase 3 clinical trials. And now recently this year we'll be announcing the addition of a commercial wing where we'll be able to take those formulations and launch the commercial products, which is very exciting for us. So given that background of the Bend Research sort of infrastructure and Dosage Form Solutions, just maybe say a few words about why we're here on the webinar with our partners at Loveless. And we feel that this is a unique partnership and has very beneficial synergies around developing inhaled drug products. And the specific aspects to that are, here at Bend, we are world experts in formulation, manufacturing, encapsulation of dry powders for inhalation drug delivery, and have proven success with small molecules, proteins, peptides, local and systemic treatment. And we'll highlight a few of those case studies here today.
And those expertise that David just highlighted from Bend Research are augmented by the expertise here at Loveless, as we're the experts in animal models and preclinical drug development. We've got over 100 animal models of disease that include everything from respiratory health to infectious disease. Working specifically with the team at Bend Research, we've got a historical relationship that has resulted in over 15 peer-reviewed publications, abstracts, manuscripts, and presentations. We have a track record of being able to do this all with compound-sparing methods in a rapid pathway from hypothesis to proof of concept and to IND enabling if the data and the project warrant. We also have a proven track record of being able to advance these internally as our own developments or with different clients in order to meet the needs of different types of partners.
Exactly, Phil. So yeah, thanks for jumping in there. That's great. Okay, so now I'd like to just take a few minutes and highlight from the Capsugel Bend perspective what our formulation platform is and how we think about developing formulations and designing the process space. And so if we look on this slide, it's sort of what we see as the components of a dry powder pulmonary platform technology. And of course, the key aspects are there's a product definition and an active. So, what are we trying to treat and what is the molecule that we think gives us the best chance of success? And then, we have tools where we look at the formulation and based on the compound physical properties, we say, okay, what's the right type of formulation for this? And there's always a component where the guidance for the formulation impact on the pharmacokinetic, pharmacodynamic performance. And that's really where the Loveless aspect comes in, because we can take these formulations and use their unique differentiated animal models and their expertise to say, if we change this about a formulation, this is going to be the impact on the PK or PD. And that's really important early on so that you get set with the right formulation, which of course is a function of the types of materials and excipients and the process in terms of, what is the morphology and what is the actual active form in your formulation. And we'll touch on each of these aspects briefly before diving into the case study. And of course, there's always the piece around characterizing the product and the analytical testing, and then the capsule-based filling process and some device. So an example of how we think about formulation development is shown here, and typically what we like to do is we like to look at some simple properties that we could measure. So for example, when you're spray drying materials, understanding the amorphous state of your active is extremely important. And then as we think about how this will behave in the lung or how to process it, we also think about things like aqueous or organic solvent for, as I said, the processing aspect. And this is just two parameters of probably an 8-parameter model that we could develop. But I think just for today's purposes, in terms of demonstrating the thought process, this should be this should do nicely. So when we look at here we're showing in this plot the glass transition temperature, TG, versus the aqueous solubility. And if we take that and we plot molecules such as fluticasone propionate or albuterol sulfate, or even some of the biotherapeutics that like peptide YY, and some other compounds that we've interacted with, we look at across this space a diversity of different compound properties, and all of these are things that people would have thought of or would like to put in the lung. And so when we spray dry these, we want to understand, okay, how stable is the amorphous state going to be? Because if the glass transition temperature is very low, the chances of being able to stabilize that in terms of it converting to a crystalline structure over time becomes much more challenging. And then we start to use this type of rationale for what types of excipients that we may want to use. And we'll also use this rationale for the type of process. So if this is going to be really hard to stabilize in the amorphous state, we may use a process that results in crystalline active. And I can speak a little bit more to that in a few minutes. So in the case where we've developed it, we've looked at the compound properties and we understand, okay, well, either from a formulation stability perspective or from feedback from our team collaboration at Lovelace, we say, well, this is fine to be in the amorphous form, or no, actually, either for stability or performance perspective, we need to have it in the crystalline form. Then we take that, we think about how to put that into a particle that's respirable, and oftentimes that leads to the need for excipients. And our rationale for the use of excipients center around either chemical stabilization. So in the case of biotherapeutics, oftentimes you want the molecule to feel hydrated, good hydrogen bonding, and so we'll select excipients that interact with it there. Physical stabilization, oftentimes if we're trying to augment a lower TG and stabilize the amorphous state, we'll add something with a bit higher TG. Surface morphology modification, putting things in such as hydrophobic amphiphilic materials that sort of go to the surface of the particle and basically create less of a resistance for particle-particle adhesion and improves dispersibility. And then oftentimes these molecules are very potent and we don't need to get very much into the lung, and so, but you still need to have enough powder that you can accurately weigh and put into your capsule and have come out in a reproducible way. And so we oftentimes have to put excipients in just in terms of dilution, and in this case we like to look at the most safe and well-precedented materials. And so that sort of discusses our thought process around formulation. And now I'd just like to take a few minutes and discuss the spray drying process as a kind of a general overview and what are the important things to think about when developing this process with respect to an inhalable dry powder formulation. And so initially we have the spray solution, which is going to be defined by the amount of solids that are present, and this also, if we're going to how we present the active to the spray solution will oftentimes determine how it's going to be in the final formulation. So if we dissolve the active in the spray solution, in most often that means that we're going to have an amorphous form of the active in the final dry powder. But as I'll talk about in a couple of slides, there are other options here where we can present nano-sized active to this in a suspension and then spray dry that for different formulation embodiments. So we pump that spray solution through an atomizer. The atomizer defines the droplet size, which that in conjunction with the solids in the spray solution will tell you what your particle size is. And then in the dryer, the extent and the rate at which we evaporate the solvent can become important when we think about things like spray drying proteins, where the protein oftentimes will like to go to the air-liquid interface, and we may need to compete that off the surface with a surfactant or other type of amphiphilic molecule. And then the particle size that we're making here is often the types of things in a normal spray drying process, say for oral consumption, that would just go through a normal cyclone. And so we've had to do some modifications to our cyclone to be able to enable high efficiency collection of these very small, very aerosolizable powders. And at the end, we end up with these shriveled raisin-looking particles that are in the range of 1 to 5 microns with a very tight span, as you'll see from the characterization data coming up. And an important aspect of developing a spray drying process, especially when you're dealing with biotherapeutics where the actives can be very expensive and very precious, is to not waste a lot of active by running empirical spray drying experiments as you're developing it. So what we have developed are a series of offline tools where, being very active sparing or maybe not even using active, we can answer multiple very important questions about the process as we're developing it, so that the first time we run to collect powder, we already have high confidence of success that that we won't be wasting active. And just to tick through some of these offline measurements, we have an offline nozzle test stand where we can bring in an atomizer, run a spray plume, and evaluate the droplet size across that, so we know right away that that nozzle is going to give us the right droplet size to achieve the 1 to 5 micron final dry powder. We also do some hot bench experiments where we look at sort of the thermal constraints of the process so that we don't see browning or baking or cooking of the formulation. And again, we can use really small amounts of active so that we don't do anything silly when we do the full-scale run. Understanding the vapor or solvent, water or solvent vapor uptake of the formulation, the excipients that you're going to use is very important, so that we don't end up spray painting the active inside the geometry. Utilizing the vast experience around spray drying and the thermodynamic operating space, we can define reasonable parameters to start with, so again that we're not going in blindly and having an unexpected incident. And then finally, as I mentioned, the cyclone, where we've had to redesign and optimize cyclone geometries specifically to collect these small particles with high efficiency. And so that's the spray dry process. And then I'd just like to take a minute here and mention up front that depending on the active properties and the input that we get from, as Phil will talk about, some of the in vivo studies that we can run, we can decide if we want the active to be amorphous or if we want the active to be in a crystalline format. And this just highlights that up front we can do everything from dissolving the API in aqueous organic solvents. We can mix that with a matrix that can stabilize it. We can also do things like nanomilling or bottom up, where we manufacture with precipitation the nanocrystal, and then all of these options can then be combined. So if we're doing combination therapies where actives may have differences in solubility or incompatible in different solvent systems, we can combine them in a mixed approach. And then the point being is that however we generate that, that's all in the spray solution, and then we can still take advantage of all of the differentiating and improved aspects of the spray dry process to really engineer the particles in a respirable range. And that's all done with all the same, there's a lot of synergy there. So what we do with the spray solution up front doesn't impact how the powders perform downstream. And finally, a slide here just to mention the equipment scales available in Bend. And we have some really custom-made small-scale equipment that enables screening of dry powders in a rapid and high-efficiency way early on. And these are some of the experiments that we'll mention specifically today with Loveless, where in using a very small amount of active and in a very rapid timeline, we can develop a formulation and work with our collaborators at Loveless to say, you know, this is going to function, and then use that to scale up to the larger scale where we're, you know, in clinical trial and then eventually commercial launch. And so with that, I'd like to hand over to Phil to take us through a little bit of the update and capability discussion for Loveless. Phil?
Thanks for that, David. And so, yeah, before I dive into anything on the case study or any of the specifics around it, I'll take a moment to give a little bit of a background on Loveless Respiratory Research Institute. Loveless is a private, not-for-profit respiratory research institute primarily focused on respiratory cures, causes of disease, and cures. To our knowledge, we are the only private biomedical research facility focused wholly in the respiratory space. We provide our services to government and industry clients alike. Loveless has two primary campuses, both located in New Mexico, in Albuquerque, New Mexico. Our corporate headquarters is located in Albuquerque itself and houses a facility of about 125,000 square feet. If you look back at the previous slide and see that we're focused in the causes and cures of respiratory health, and you think about the relationship between our two campuses, our North Campus, as we call it, or our corporate headquarters, is our basic research facility. It's that facility and the group of scientists at that facility that are looking at the causes and understanding the disease within respiratory health. Our South Campus is located on Kirtland Air Force Base here in Albuquerque, New Mexico. That facility houses about 325,000 square feet of labs, and it's that facility where we do more of the applied research. We're looking at, after we understand the disease models, how do we intervene in a prophylactic or a therapeutic manner and understand and be able to treat disease, whether it's screening for new compounds or understanding what different changes in environmental exposures might alter a disease state. These two campuses bring together a very diverse range of scientific disciplines required to complete these types of studies. As you can see listed on the left, there's a broad range of everything from aerosol scientists, analytical chemists, radiochemists, radiobiologists, immunologists, pathologists, and folks specializing in toxicology. All of those disciplines are needed for the Loveless team to be able to apply these animal models, to develop them, and be able to support our clients and our partners in understanding their studies. One of the important aspects that I think is well highlighted with the case study we'll present, and that David has talked about a little bit, is the translational nature of the research we do here at Loveless. As I mentioned, we have a group that's largely focused on understanding the basic mechanism of disease, understanding lung disease. We then have teams that work collaboratively together to try to develop and implement animal models that mimic that clinical disease so that we can understand the natural history of the disease in an animal model, compare it to what we know about the natural history of a disease in a clinical model, and then develop treatments or other models in the preclinical arena that allow us to screen therapeutic compounds. In this case, we'll be talking through with an example on insulin. More importantly, we're then able to work together to take those compounds after we've understood their efficacy to be able to understand their safety and transition back into clinical studies that have real relevance. We have a proven track record of being able to do this in all sorts of different pulmonary disease indications, from pulmonary fibrosis, influenza, COPD, and pulmonary hypertension. As I showed that previous slide, those images make the job of going from understanding disease in the clinic, treating it preclinically, and moving back to the clinic look pretty easy. They're bright colored pictures. This slide is one of my favorite slides, and it really shows why we need that diverse range of everything from aerosol scientists to toxicologists to be able to make those bridges. These are lung casts of different species that Lovelace has research working with. If you look at the human on the right, all the way down to the mouse on the left, those pictures should show the different complexity of the aerosol and the quality of the formulation that we need in order to deliver an aerosol across different preclinical species and have the ability for that formulation to have meaning when we move back to the clinical setting, hopefully as the efficacy data and the safety data warrant. So it's working across that continuum with good partners like Bend Research and being able to generate those formulations that really enable the success of any program, as I said, as you move from that preclinical space back into the clinic. The current study that we're going to talk about today that David and I will go into some detail on is a case study where we looked at a product that at the current time was marketed, inhaled insulin in the form of Exubera, and a formulation that the Bend Research team spray dried and developed that we'll call a dextran 10 inhalation formulation of insulin. The goal in this model was to look at the pharmacokinetics and the pharmacodynamics of orally inhaled insulin. Specifically, we wanted to compare and contrast both of those. Once we had the data, we looked at modeling the arterial blood insulin concentration with standard pharmacokinetic methods using Win non-Lin to try to see if there were differences or if we had formulations that were essentially equivalent. So now I'll hand it back to David to go ahead and talk through some of the technologies that his team utilized to make a formulation that enabled this case study and this preclinical study here with our teams at Lovelace.
Great, thanks, Phil. That's really interesting. I too really enjoy that slide with the lung cast. It's amazing. So now just to take a few minutes here briefly, because the real interesting thing here is going to be, as Phil takes you through, sort of how this dry powder interacts with the biology and the models that Lovelace came up with to kind of demonstrate that. So I'll just take a few minutes here and highlight what we made and how we characterized it. But essentially, as Phil mentioned, for this specific study that's in the PharmSci Tech journal, we were looking at higher molecular weight polymers with hydrogen bonding sites to help stabilize the insulin and also give a really high-performing product because of the high glass transition temperature. It avoided adhesion between the particles. And we were able to do this very rapidly with a very small amount of API. And we started with our customized Bend lab dryer, which, as mentioned in a previous slide, sort of spans the scales from about 200 milligrams of powder up to hundreds of grams, and of course that depends on how much solid you can dissolve in your spray solution. But effectively, the purpose here was to make and collect particles that were around a mass median aerodynamic diameter of 2 to 2.5 with a GSD value on an impaction test of 2 or less. And using all the tools that I mentioned on a previous slide, we mapped out a process space and then used that to select these conditions. So here just highlight going through the drying gas flow rate, atomization, feed rate, the solids loading, which is about 2 weight%, and then the inlet temperature, which seems high for biotherapeutic, but actually because of the evaporative cooling that happens right away, the product really only sees the outlet temperature, which is around 55 degrees C. And as discussed in the paper, which we don't have time necessarily to go on here, the 55 degree and those process conditions were selected because we knew and had measured the glass transition temperature of the dextran much, much, much higher than that, even at the relative saturation of the dryer here, which I think was around 4 to 6% relative humidity. And so that outlet temperature was such that it's much, much lower than the glass transition temperature of the product we were making, which means that we wouldn't have issues with sticking of the product, but also that the dry powder was essentially frozen and not mobile, meaning that we felt confident that that would hold the insulin in place at elevated temperatures and not allow for any sort of chemical degradation or unfolding. And then in this case, we were looking at approximate batch size of about 7 grams, and at this small scale getting a yield of 70%, which typically as we scale these formulations up, we start to get higher into the 80 and even 90% yield. And we did perform using a Malvern dry powder method, a geometric particle size, and showed that we were getting about 2.6 micron particle size. And so when we tested this material, and here we'll talk a little bit about some of the methodology. So we looked at dry powder impaction, and the way these tests are run is about 10 milligrams of powder is put into a Capsugel VCAPS Plus special grade for inhalation, which means it's an HPMC capsule, so it excels at the lower relative humidity storage conditions that are required for these, typically for these inhaled dry powders, meaning that we really like to store these at less than 10% relative humidity, which for gelatin-based capsules is not an acceptable condition. You get cracking and brittleness. The VCAPS Plus inhalation grade also has a specific and lower, very small amount of lubricant, which enables or basically enables dry powder formulations because you don't have the excessive lubricant that then interacts with the dry powder and causes powder hang up in the capsule body. And so that capsule, these were filled into those capsules, which were then put into the off-the-shelf Plastiapi monodose inhaler, and then fired into an NGI at kind of standard conditions, which are listed in the fine print below. And as I said, this has all been published, and it's all in the paper. If anybody would like more detail, they can find it there. And so what we found was, as we expected, a high emitted dose. The fine particle fraction less than 4.6 microns was very high. The calculated dose delivered to the lung was very high, and we achieved sort of our 2 micron mass median aerodynamic diameter and geometric standard deviation. And then, this was work that was done here at Bend to make sure that we weren't sending to Lovelace anything that we didn't think Phil and his team could handle in terms of the animal model. And then once at Lovelace, they, with their aerosolization methods that were more appropriate for the model Phil will talk about, they confirmed these results. We also like to look at dynamic vapor sorption and how that relates to the glass transition temperature. And this is used to define the storage condition, because if we take on too much water, we're going to have stability issues both with particle-particle agglomeration as well as enabling mobility of the active in the particle, which can allow for unfolding or even aggregation in the dry powder state. So we use this to define the storage conditions, and we typically like to be less than 3 weight % water, and so we use that to define, how the less than 10% RH. And then this is just a beautiful scanning electron microscopy image here showing the really shriveled high surface area, low contact point of the spray dried powder, which means less contact between the particles means less van der Waals interaction and higher dispersibility for a well-performing dry powder product. And this slide was just a point to say that in this paper we had evaluated dextran 10 as a superior performing excipient, but it's not precedented, and the safety does need to be demonstrated out. So we have also shown that you can develop these formulations at different loadings with the same process platform by substituting the dextran 10 out with a smaller molecular weight sugar that's a bit more precedented in the industry and showing effectively a well-performing product. Okay, and with that, I think that's a good summary for the dry powder that was sent to Lovelace, and now we'll let Phil take over here and tell us what they did with that dry powder. So thanks, Phil.
And so thanks for that, David. And David did a great job of describing what his teams will do to develop a dry powder for inhalation. And as he described, a lot of the methods that he detailed apply more to a clinical setting. And in another webinar, on another day, and another presentation, we can go over how a patient will take that device and that formulation, and quite frankly, they'll use it sometimes correctly and sometimes incorrectly. Let's assume for right now that we wanted to do a preclinical model off of how a patient would use it correctly. In general, a dry powder device is something that a patient should be trained how to use. During that training, they should be taught how to do a nice, calm, controlled inhalation over between 1 and 3 seconds that fills the lungs completely. Immediately following that, they should do generally a 10-second breath hold, followed by a calm exhalation. Whether or not people are successful at doing that, again, like I said, that's another story. But as we roll that over to a preclinical model, whether that's a large animal or a small animal, the ability to teach one of those preclinical animals to do a nice controlled inhalation, get them to do a breath hold, or a nice calm exhalation is difficult at best. So what I'd like to start out with talking about is how the team at Lovelace, many years ago, in fact working on Exubera, developed methods to be able to try to mimic a clinical maneuver so we could take well-engineered dry powder formulations like the material that Bend Research develops and be able to transition them into a clinically relevant preclinical model. In doing that, we developed a system here at Lovelace that we've called the dry powder bolus delivery system. If you look at the image on the right, this system consists of a dry powder reservoir, an expansion holding chamber, and an endotracheal tube. And the forced maneuver that we perform is actually performed with a positive ventilation bag, an ambi bag, just like you would see it in a clinical setting. In this model for the animals, we anesthetize them with topical isoflurane. After they're anesthetized, we intubate them with an endotracheal tube. Following that, we induce apnea by overventilating the dogs between 10 and 15 times. And for those of you that aren't toxicologists in background, what this does is this over-oxygenates the animals. So the animals' internal regulation system that determines how often they need to breathe stops. So the animals are no longer breathing. This allows us, and the teams here at Lovelace, to be able to breathe for the animals. In doing this, we aerosolize the material from the dry powder reservoir into the holding chamber. Immediately after we deliver that aerosol into the holding chamber, we do a forced maneuver to the dog that is not breathing on its own and deliver that aerosol to their lungs, followed by about a 3-second breath hold. While we can't get a perfect clinical maneuver with this, there are some subtle differences. This maneuver allows us the best opportunity to take well-engineered materials and do a clinical type maneuver to these animals. So not unlike what David and the Bend Research team did in terms of in vitro testing, the team here at Lovelace received the Exubera materials and the Bend Research spray-dried insulin material, and we had to work through our in vitro characterization to make sure that our results matched what the Bend Research results were, and then we're able to transition those over to the in vivo side of the testing. Two of the major points that we look at in terms of this particular delivery system, we look at the efficiency of the material to be ejected from the reservoir, and we look at the efficiency of the material to be delivered to the terminus of the endotracheal tube. We also look at particle size throughout this model. And in doing this work, we look at a range of different loadings and some subtle different variations on the delivery technique in order to optimize that delivery efficiency. And you can see here in this table, we were able to develop a pretty highly efficient system to deliver this material both out of the dry powder reservoir and to the terminus of the endotracheal tube for that material to be presented to the dog's lungs. We'll go through and talk about it in a little bit, but our goal here was also to match the dose of insulin, knowing that there were different blend percentages within the formulations, but to match that delivery of insulin to the terminus of the endotracheal tube. So we had a good PK/PD study. So with regards to this specific model, it should be noted that all of these tests were performed under the Lovelace Institutional Animal Care and Use Committee. That's our IACUC committee. Many people use that as short. And that we're an ALAC accredited facility. So all of these procedures were performed according to those procedures. This particular model requires a surgical implementation of an arterial catheter prior to the model. So we used our veterinary surgeons to implement that, allow the animals to recover, and then on the day of the study we implement catheters, intravenous catheters, for glucose infusion and in order to maintain euglycemia within the dogs. This study was done as a 4-animal crossover study with 2 animals receiving each formulation on each day, so it was a full crossover. Within this study, immediately following the inhalation, we collect 14 serial samples of the blood to look at, of arterial blood, to look at insulin, C-peptide, and blood glucose. Within the model, we utilize somatostatin to suppress endogenous glucose production, and then we infuse a constant rate of glucose to maintain euglycemia within the animals so that we can completely understand and appreciate when we do our data analysis that the insulin we're quantifying and that we're modeling in our pharmacokinetics was from the oral inhalation from the Bend research material and or the Exubera material that was delivered via the inhalation maneuver. So the first thing we always want to look at when we look at one of these studies is what dose did you actually deliver? So for the dose delivery in this study, these were all normalized to individual animal body weights taken immediately before the doses. You can see we gave the exact same dose of insulin from each of these products. That enables us to be able to read through the data that I'll present in a little bit, and like David has mentioned, is presented in the manuscript. So we're starting from the same foundation in terms of delivered insulin dose. The first data point we look at is C-peptide. C-peptide is a measurement of endogenous insulin secretion. So if we have suppressed endogenous insulin through somatostatin, we should see these drop very rapidly and stay at or near the limit of detection of our assay throughout the duration of the study. As you can see on the graph on the right, this is exactly what we saw for both formulations on both days of this crossover study. That allows us to read through the data, and as I said earlier, we're able to ensure that the insulin we're quantifying in the arterial plasma really came from the oral inhalation from the dry powder formulations. Because of that, we're then able to look at this graph. We're able to look at the graph on the right, which is the arterial plasma concentration of insulin for each formulation as a function of time. So you can see both of these follow apparent first-order elimination. And I think more importantly is that you see that how quickly the material was absorbed into the systemic plasma and how high the Cmax and the Tmax for both of these appear to be very, very close. And we'll get through some of the modeling in a little bit. The other thing that I think should be noted is if you look at these graphs, it does appear that the Bend Research Dextran 10 insulin material appears to have an increased insulin concentration from about 30 to 35 minutes to maybe about 90 or 100 minutes later. We evaluated the statistics on this, and while it does look like there's a trend there, that was not determined to be statistically relevant. The next thing we look at is we look at our blood glucose and our infusion rates to make sure that those were similar across all the animals and across all the formulations. So as we maintain euglycemia, as you can see in the top graph, our arterial blood glucose levels are constant, and the infusion rate required to achieve that was not statistically different. And then we get into the pharmacokinetic modeling. So as you see here in this table, probably the most important part of the pharmacokinetic modeling is the Tmax, the time at which the maximum concentration is achieved. We're very close for these two formulations, with a Tmax of 14 minutes for Exubera and 20 minutes for the Dextran 10 insulin formulation. Coupled with that is what's called the Cmax, the maximum concentration achieved in the systemic plasma. As you can see from the table, they're both at about 120, 125 microunits per mil, meaning that the speed that each of these formulations were absorbed and the completeness were very similar in terms of how quickly they were absorbed. That speaks to the quality of the formulations and the similarity between the formulations in this inhalation delivery, and that they were likely deposited very homogeneously throughout the lungs of these animals. Reflecting back on that concentration time profile for the arterial plasma insulin, if you remember I talked about the fact that the Dextran formulation, the Bend Research formulation, had a slight tendency to an increased concentration throughout those middle time points. That's also reflected in the area under the curve, as you see on the bottom line of the table. You see a little bit of a tendency for the AUC to be higher for the dextran formulation when compared to the Exubera. However, as I noted earlier, that was determined not to be statistically significant. So with regards to this specific case study, this shows the ability of Lovelace and Bend Research to work together to assess, in this case it was a proof of concept formulation, in a PK/PD model where Bend Research was able to develop a novel engineered spray-dried inhalation formulation and hand that off to Lovelace. We were then able to move that into an in vivo PK/PD model in dogs and evaluate the bioequivalent or the equivalence of these compounds in the PK/PD model, looking at both some PD endpoints and some of the PK modeling. And I should mention that this has been published in a recent AAPS journal in an issue focused on inhalation formulation with guest editors that had several other articles focused in this area. As we transition away from that topic a little bit on that particular case study, it's important to note that, as I mentioned earlier, that Lovelace and Bend have had several other examples that have been published in the literature of our ability to work together. Now I'd like to go through a couple of those examples and talk about some other different compounds and different disease models that have been able to show these synergies in. The first one utilized a small molecule in a dog model of bronchoconstriction. In this model, we looked at PK and PD. Our PK data are shown in the table on the left. For this particular material, this was an albuterol dry powder and an albuterol nebulizer. And you can see that the Tmax and Cmax are very close between those two formulations, and the half-life is very close. As important was the pharmacodynamic readout, which is a measurement of bronchoconstriction. With each of those bars, the closer each of those bars are to 100%, that means that the formulation completely stopped bronchoconstriction in this animal model. This has been well published and has been, this particular data set was published in Pharm Research as shown in the reference on the bottom of this slide if you have any other further questions or would like to look at it more. But this, like I said, this showed the synergies of Bend and Lovelace in a small molecule for local delivery in bronchoconstriction to the lungs in a dog model. Another example that we'd like to touch on is an example of a PK/PD model that we did in rodents for a systemic indication. This indication was appetite suppression. In this model, we utilized pharmacokinetics following delivery by intraperitoneal injection, subcutaneous injection, and inhalation delivery to model the systemic pharmacokinetics and compare what we determined to be an equivalent formulation. So on the left, you can see that following a dose of 0.1 mg/kg by IP or sub-Q, we see similar systemic pharmacokinetics. Through the different doses and different modeling of the pharmacokinetics following inhalation, we determined that a dose of 0.2 mg/kg by inhalation resulted in similar systemic pharmacokinetics. So our hypothesis was then that if we moved that dose over to the efficacy model of appetite suppression, we would see similar pharmacodynamics. And as you see in that graph on the right, the dose of 0.2 mg/kg by inhalation showed similar efficacy in this appetite suppression model. These data are in press right now in drug development and industrial pharmacy, and I would encourage anyone who would like to grab that article and review this more in detail. A final example that the Lovelace and Bend Research teams have worked together on was a small molecule that we did a PK assessment for a lung cancer treatment model. In this method, we used a novel technology by Bend Research where they actually made a nanocrystalline material for Lovelace to use in a rodent PK model. We were using these data to look at bronchial alveolar lavage, fluid concentrations, lung tissue concentrations, and systemic plasma concentrations following IV delivery of canthothecan and 2 different inhalation doses of the novel dry powder for inhalation. Our goal here was to show similar exposures in the lung tissue while reducing systemic exposure, with the goal of being able to transition this into an efficacy model where we could utilize, in the same manner as we showed with PYY with the previous compound, to be able to show the differences between the dose that you would need to use to see similar concentrations for efficacy. Hopefully, as we transition that through, we'll be able to show some efficacy data. And last, I think we should note a couple of things that the Bend Research and Loveless team have that they're working on currently that hope to have publications on coming in the near future. One is a PK/PD model where we use a small molecule to assess the differences in formulation. Many of the things that David described earlier that his team has the capability of modulating and controlling, and seeing what their impact have on systemic PK and acute pharmacodynamics in a large animal model. And also we have some work going on with a small molecule in order to assess its efficacy in an inhalation delivery for lung cancer. So with that, I'd like to transition it back to David, who will inquire and hopefully be able to address some questions and be able to wrap up our webinar today.
Okay, great. That concludes our presentation. I see that we have several questions, so let me get started with the first question. And the question is, could you please explain stability of the engineered powder? And that's a really big question, and we could probably have an entire webinar solely on that aspect. But when we talk about stability of the engineered powder, typically because we're using the spray-dried process, what that means is we're going to have amorphous material. And there are two aspects to the physical stability of that. One aspect is, the state of the active. So if we've made the active in an amorphous form, we want to keep it amorphous. And then the second aspect is we've made these, elegant, small 2-micron particles because we want them to get into the lung, and we want to make sure that they stay at the 2-micron particle size on storage. And both of those aspects are going to be handled by thinking about the glass transition temperature of the matrix material or the surface material of the particle. So in a really short summary of that question, and happy to talk more offline about it, the glass transition temperature of the material is going to be very important, and that's going to be a function of the material you choose as well as your storage condition in terms of the relative humidity. So hopefully I answered that question sufficiently. Let's look at another one. Okay, here, how much material is required to complete a PK/PD assessment similar to what is shown? Phil, would you like to answer that?
Yeah, David, thanks. I think that's one that I can take. So in general, one of the most important, there are two real things that contribute to the material needs for one of these studies. Those two things are the efficiency of the delivery system, that speaks to the delivery system itself, and to the quality of the material that's being delivered. The other thing that contributes significantly to the amount of material required is the target dose. So if you refer back to the slide that I presented with regards to the dose delivered to the lungs of these animals, we're at a 0.12 mg per kg dose. For this program, this entire program, the aerosol development, the testing, and the actual animal dosing, this program required between 50 and 100 milligrams of material to conduct all of this. So obviously based on the different, and sorry, what I should have said is this was a fairly efficient powder when you looked at the delivery efficiency presented to the end of the endotracheal tube and of the amount of material that was ejected from the dry powder reservoir. Because of that, our material needs were relatively low. As you can imagine, if you increase the dose significantly, there can be some increases in the amount of materials consumed and needed to perform these studies.
Okay, great. We have a few more questions. The next question says, I noticed that Tmax for Exubera was substantially faster in your model than in actual clinical use. Is that typical of your model? Phil?
So with regards to the Tmax differences between the clinical model and the preclinical model, we haven't specifically looked at, or I can't say that I specifically have looked at the differences between what is seen in the clinic versus our model. What could contribute to those things could be the differences in the inhalation maneuver. There could also be differences in the deposition pattern within the lungs. As you can imagine, there's variability around each of the different maneuvers, as I talked about, and there's differences in the lung itself. The other thing that could contribute to some of those, I don't know the studies specifically, but you might want to look at what blood sampling time points. If you go back to the time points that we selected in this study, we're pretty heavily weighted on the front end in and around that 5 to 35 to 40 minutes to try to capture that. So it's important to also look at those if you look at both the clinical study and the preclinical study and making those comparisons. However, most of these materials, the dry powder materials that Exubera itself and that Ben Research have used, as David mentioned, they're largely amorphous materials, which provides an opportunity for pretty rapid absorption, resulting in fairly quick Tmaxs and pretty high Cmaxs when we look at systemic availability.
Okay, excellent. Looks like we have time for maybe one more question, and we'll follow up with other questions if we don't get to them offline. So the last question is, on the powder engineering, why did you choose to use trehalose instead of inhalation-grade lactose, which is the standard for DPIs? The answer for this one is fairly simple. In the case of insulin, we have some free primary amines and some secondary amines that we didn't want to have interaction with in terms of a reducing sugar and have a browning reaction. And so oftentimes for biotherapeutics, you're sort of forced to use a non-lactose excipient, and trehalose is a reasonable substitute for that. So those are all the questions we have time for today. If you have follow-up questions, please feel free to contact us directly via email. And as I said earlier, questions that we weren't able to answer in the timing of this webinar, we'll get back to offline over email. We do plan to host additional webinars on our array of proprietary technologies and hope you'll continue to participate. Our next webinar will be held on June 3rd and will cover our biotherapeutic formulation and processing capabilities. Please check the Capsugel or Ben Research website for details and to register. On behalf of Capsugel Dosage Form Solutions and Loveless Respiratory Research Institute and you, Phil, Dr. Cole, I'd like to thank everyone for joining us today. Please fill out the survey that will pop up on your screen as soon as the webinar is over. Your feedback will be incorporated in a future webinar plan to make sure that we're continuing to have material that's of interest and relevant to the audience. And please contact us at the email address given if you have any questions or comments, and we will follow up. Thanks again for your time and attendance today, and hope everyone has a good rest of their week.